Hello, everybody. So it is Saturday. My cold sore. I think this is the worst it's gonna get. And then some of the stitching came off, just dangling in my mouth. I worked earlier, but I got a 2-day weekend as usual. I was working every day for the last few years, but then I did take a year off for that last one right before they kicked me out of chiropractic school. But I did work earlier, so I got a weekend coming up, starting tomorrow. I just started drinking this beer. I fixed my car, getting a root canal Tuesday. I'm getting 2 root canals actually. I think I just had a tooth pulled that was a root canal that was infected. And then the tooth next to it was cracked, so they had to get that ready for a root canal. I'm getting that one, a root canal. Then they're going to do a bridge to go over the old gap that they put a bone graft on for that removal of the infected root canal up on the top jaw. And, in 2023, I had glycolic acid fillers, and they kind of got rid of the little cat whiskers, that show whenever you're like somewhere and it's like this and you got like little whiskers, like they call dark. So I had them in, and actually they were still lasting. They were only supposed to be there for like a couple of years, and they were in there. But see, like, I think I got because I was taking antibiotics, it weakened the viral latent inactive stages. So for the herpes simplex 1 cold sore, the Novocaine had an interaction with some of that glycolic acid, and it set off some reactions that said, oh, you guys go fight it out. There's no bacteria controlling, mediating thing. So then the herpes simplex virus 1 decided that it was going to start activating itself. And then I got this. I haven't had a cold sore in like 5 years. So this really sucks. And it looked like I had like a fat lip, like that duckbill lip, not even the one that's cute, like where your lips like filled, it was like partially filled, but like a duck lip right there. And it's kind of filled like the whole thing. And I think it was the Novocaine leaking out because it actually felt numb too. Because I've had cold sores before and they're usually just painful and dry, and then they crack. It did that, but it wasn't really that. It's not painful. It was like numb. So this time we're going to go over the colon cancer that we've been working on, and everything was the top gene. We explored KEGG last time. It is where you go for the gene variation analysis or gene studies, variability analysis. There's a specific name for it. We don't really need to know that name because that's not really what we're doing, but we're just comparing genes. Like everything that we're doing, there is a name for it and there's a method. I might be going in and out of order, but it's kind of basically the same thing. And we're just getting the top genes. We're finding these target genes. We realize that there's actually a bunch of target genes, and they have a knockout method to inhibit those cancer genes right now. The thing is, they probably don't have ways of really satisfying the human health, with organs being able to handle all that substance, like the liver, kidneys, your heart, so they might be able to knock out those cancer cells, but at the same time, they might kill the person by giving them organ failure. So it is good to know that they have things there, and this is what it leads to. You're analyzing these genes, and then they're finding ways to see if there is a network, a link analysis between them. When you have these downregulated genes and these upregulated genes, the downregulated genes could be the bad ones that are interfering with the immune response, or maybe giving you too much immune response that's like too much for your body to filter at once, like lymphoma, where the B cells, there's just too many macrophages, too many B cells, too many white blood cells, and they get clogged and build tumors, and they're not the right type, so bad ones get replicated. And then and then the good ones upregulated, they could be upregulating genes that actually have nothing to do, genes, okay, so like tumor suppressors, we want those upregulated, right? So they can go out and survey your body and make sure that you're not forming tumors, that cells are good and they're not getting replicated, right? And then you got the upregulation of genes that are usually inhibited. They're probably upregulated because they need to make a whole bunch of them. Like that's what happens when you have low blood iron. Your body needs to make more blood, or when you run out of a lot of blood, you have anemia, low iron anemia. And so it makes basically inadequate, not well prepared blood cells to get at least some oxygen to where it needs to go. But you can't live that way because they need to start making the healthy ones. But when you're stressed all the time, it can't. Sorry, I think I'm just disturbed by the look of my cold sore on my face. It keeps interfering with what I'm thinking, my trail of thought. So it looks like Timo always sends me stuff. I swear I haven't bought anything from them, and I've never spent more than $300, but I haven't bought anything from them since like maybe December or November. And yeah, I keep getting stuff from them. They actually have pretty decent stuff. Their fashion stuff, though, it's pricey for those dresses, and I would say that they're not really that fabulous. You know what I mean? I ordered a dress, and the arms were, of course, too small for me because I have larger arms than most people. And I don't really care. That's my body size, so I'm over that. But I don't like having a cold sore. I don't like that. Okay, so I'm going to share my screen, and then we're just going to go to the AURKA's site to look at where we went. Actually, I'll share that in the details and the chat. So I'll just go ahead and share the entire screen. And this is where... All right. So we went to this one. This is what we were doing. And it's basically just all part one. It's all in part one. Same with autism. And there's multiple parts in it, but it's all from part one. And then we're just leaving off where we had all these genes, and all of them scored exactly the same across in the predicting test and the holdout validation set. These genes right here, different genes in each transcript per million, fragments per kilomillion, and then the read counts. You got the read counts right here, fragments per kilomillion right there. So I'll just go ahead and I'll share that link here. I didn't share it in the details yet, but I will. All right, and then we're going to go ahead and go to the ARPubs document and then get ready for a bunch of reading. All right, we're going to go to this one right here. This is what we were working on. This is our markdown file. And then, yeah, so we'll just run all the chunks right now so that we get where we're at. It doesn't take that long to run this. We're not doing a major analysis here. I mean, it is, but it's faster. It's not like we're doing clustering where it takes like an hour and a half to do anything, or using Sirot where it takes like 20 minutes for each one, each of your samples. Let's get rid of this, that's not where we're at. So we're just going to look at the last data frame where we what's going on here? Okay, so we're just going to go ahead and, oh, I thought we made okay, I guess we didn't make a table of all genes. We get the top fragments per kilometer, hits per million, and counts. But let's go ahead and, because we did the machine learning last few days ago. And here's all of our, okay, everybody see this is where it is. Data top 3 sets. We did do that. Okay, great. That's what I thought. Data, 60 observations, data top 3 sets. Let's look at that one. So we'll just look at it in the viewer. Here's our gene symbol. We're going to go over to genecards.com because we want to know what these genes are doing. So we're going to go ahead and arrange. They're not ordered. I don't think, yeah, they're not ordered. So it looks like we can order this, and then we're going to see what all of these genes are, inhibited or not. So we can just go ahead and Dallas, get down. Stop that. No, go, get down. Yeah, we're going to go ahead and just open this up in Excel and add these manually because that's just better for it instead of just writing a whole vector for it. I mean, yeah, because it's just easier. It's more practical. So we already, I don't believe we wrote this out though. So let's see. No, I don't think we did. So we're going to go ahead and write this out. And then I'll share it. Okay, so this is part 4, I think. It's like day 4. On this project, we will write out the data top 3 sets, data frame to CSV, open it in Excel, and then look up each gene after we order it in decreasing order. Okay, so let's go ahead and go data 60. It's going to be ordered, so we're just going to go data top 3 sets. We're going to order data top 3 sets by gene symbol. Decreasing equals true, comma for the columns. And then we'll just write it out. CSV. Write .csv data 60. We're just gonna call it data 60. Ordered top genes .csv names equal false. F for false. We'll just go ahead and run that. We can also look at it in our viewer just to make sure. Oh, we used it by gene symbol. Oh, okay. So there, you know what? There are some duplicates. So some of these genes are in other ones. So we'll just go ahead and leave it like this. We actually wanted to do it by the expression value of fold change. But we can do it this way. This is the way we know that we don't go over these genes too much. And it's okay to keep the duplicates because we want to know. Oh, well, it is still the same value. So actually, let's not keep the duplicates. Let's go ahead and get rid of the duplicates. So we're going to keep this the same. We're just going to make it different. Gene symbol. Okay. And then let's also, no, we're going to not order it. We're actually just going to see not duplicated. Data top, shoot. Data top 3 sets by gene symbol. Okay, let's see how many are in here. Whoa, there's only 22 genes. So a lot of them are the same genes, which makes sense because it's fragments per kilometer, transcripts per million, and recounts. So yeah. Let's go ahead and order this now. We'll just keep it ordered by the now let's not keep it that way because there's no duplicates in it. But now we're just gonna keep it's gonna be data. Now it's gonna be data 23. There's only 23 unique genes. So data 60, order data 60 by let's do the fold change. I just want to do by counts. Decreasing equals true. They should be the same all around. And we'll just go ahead and keep the data 60, but we're gonna go ahead and write .csv data 23. Doesn't matter if you do quotation marks or single quotations, but I'll just keep it the same. Data 23, unique 60 top genes.csv, row dot names equal false. Okay. And then let's just look at these 23 genes. And then I'm going to upload it to Google and share it with a link. We're just going to look at the genes. Which genes are there? Well, that's cool. We'd only have to look up 23 now. And let's look at the data 60 by gene. Dang it. I get the cursor interferes with my viewer. It blocks what I'm typing. Oh, right. Yeah, because actually data 60. Yeah, because we put not duplicated. Oh, I see. So actually, let's just go ahead and get rid of that one. And then we'll get rid of this one too. And close that out. We'll go ahead and save it. And then let's just go to our files. And then we'll go to the newer ones. We'll just delete that. Great. And then we are going to open up this one, but I'm going to upload it to Google right now. Google Sheets. All right. So usually when I press this, I get a list of a bunch of stuff that shows, but I guess apparently not. So I'm going to just go to Sheets. Okay. My security camera was so annoying. It doesn't let me clip a timeline and download everything. I have to go into every tiny little clip over 3 hours and download it and wait just as long. It's like I'm watching 3 hours of stuff to do it. It took me 2 hours to download from my own security system some clips. And it's not every clip either. It's only with movement. All right, let's see. We're going to go ahead and add this file, and I'll share the link. I have some taffy. I'm going to eat some of this. Actually, I'm uploading a video and doing a live stream and running our what is that real deal? $4.91. I've only been doing this. We can explore. I have some PDFs. I've done many different things, but of course you don't remember it, but you get better at whatever you do, that you do it all the time. And you can add a tiny bit to it while you're doing it. Okay. I'm going to go over here. All right. After we manually find each gene summary, we will save it as a file and upload it from this folder that we will save to as a different name, so that it doesn't rewrite our work when we rerun this script. Okay, so we're gonna go ahead and open it up. We're gonna leave this for a bit. Let's go ahead and save this. This is what we named it as. We're gonna just put summaries added. Let me open it. All right. We're going to save as, and it's going to let us. Summaries added. Okay, so here we are. Right here it says that. Let's just go ahead and look at what we got. 23 genes shouldn't be a problem. So in the details, I did say that we're going to look at the KEGG also and possibly look at other genes, because it occurred to me when I was chatting with somebody while I was doing massage for my day job, that there is a possibility. What if there is a connection between the newest healthcare techniques for pregnant women, because ultrasound is the safest. It doesn't have any kind of ionizing radiation. And so with MRI as well. So what if all these kids that are born with autism, because there's a lot more being diagnosed, though in the Merck manual it says it's due to better criteria, qualifying criteria to rule in autism and not something else. What if the developing brain in the embryo is affected by ultrasound, because the ultrasound goes up in there so close to the developing embryo. It starts from that little, merger, and then it turns into like a blastocyst, and then it keeps dividing, and then it separates, and then it forms all of these different things. And then there's like a whole bachelor's degree in the developing child developmental psychology. What if that brain wasn't okay? They said they ruled out vaccinations already, but what if ultrasound affected? Because it does on the spectrum, but not a vibration. It's a high frequency. They got it from whales and dolphins communicating. They don't see, they only look at for communication. Although autistic people can see, but you never know, because it is a new development, and maybe that is a side effect of ultrasound, is that some embryos will develop autism. Don't know. Came up. I thought of it. Maybe someone else had that little thought, right? And I had 3 years of chiropractic school. Yeah, I don't remember all of it, but I remember a lot of it. A lot of it for people to say, like, wow, you know a lot. That is a lot of it. Wow, how did you? And I'm like, yeah. So that's how. Okay, so that's the thing. We're gonna look up on KEGG the developing brain, see what genes come up, and then we'll see if we can find anything for ultrasound too. Maybe we'll have to look at the NCBI. This could be a next project, coming up with something. We might start taking this outside of our original project, which is the Epstein-Barr virus associated pathologies. We're going to keep that one going, but little side projects. We'll just come up with little things that we can use our data science skills to go extract information to answer a question that we develop. We would develop a question, come up with a way to prove it, disprove it, find features, information, merge that information, join the data sets, see what kind of classification or what kind of regression you want, or unsupervised learning with clustering, so that we can use to say, maybe there's enough information here that there is possibly an association. What do they say? That's one of those things they say that's like, it's common sense. It makes sense. Correlation doesn't mean causality, because like they might be correlated going in the same direction, or negatively correlated opposite, only opposite when they're in each other, but does it mean they're associated? Yeah, so anyways, let's just go ahead and put that there. We're also going to look up some other genes, because vertigo, one of those things, they just say control with diet. Maybe there's some genes in there that are highly expressed when someone has a like a flare-up, I guess, of that. Same with fibromyalgia. So what everyone should do is, like, when they are an adult, they should really just take, like, whenever they are perfectly healthy, sleeping right, hydrated, on a great diet, nothing in the world bothers them, happy, content, not overly happy, not overly frustrated, not like bipolar, not highs and lows, but totally where they want to be, and nothing bothers them, nothing got in the way, nothing stresses them out. They should get their full genome sequenced. And then, just like stem cells, keeping them in the freezer for another day, that you might be able to use that for your own genes to use to build your failing body to make it better. They should also look at that and compare it to when your body starts to break down due to whatever. And see, because then everybody will have a control sample. Okay, they'll have a healthy baseline. And then you can look at it, and it could totally be personalized healthcare, because that's what they want. That's the model. It could be a business model. It is a business model. Now healthcare is all business. Yeah, it's all about profits. Yeah, it's about making people better, because obviously you want to do that, but it's always about the bottom line, as they say. So, yeah. And it's actually, it's quite thankless. I really, because nobody wants to have subpar treatment. That's less than perfect. They're not happy. It doesn't matter what you did to fix them to make them better. They're not really better. We have to sacrifice some of the part. So let's get started. We're only into this, I would say, about half an hour. Yeah, I was right. 28 minutes. 30 minutes. Oh, cool. Look at that. So last time when I flipped out on my neighbor, he was being stupid. It cut out me, and it said 2 hours, but it cut out me, and I said an hour. So whatever. That might happen today. I'm not sure. It's Friday, Saturday. And this is how far. It's a 3-hour video, and this thing has been uploading for like an hour and a half. Forever. Okay. Are you guys excited? So before we start this, let's go ahead and put a column here. I shared this with you. Okay. We don't want to edit this in here. Okay. So I'm going to cut this. We're not going to add a column here. I almost did. Don't do that. We're going to go to our regular one that says summaries added. And we're just going to hide everything except for the gene symbol. And then we'll just insert a column. Insert. No, actually, it's going to be at the end. So insert a column. And we're just going to name this gene summaries, gene card summaries. Okay, so our first one, let's shrink this down. We don't need all that. And we're going to go ahead and look up our first gene, but we are going to go here. We'll just make it a separate window because I'm sharing my entire screen. So we'll lower this. You can see it, right? Okay. Nope. Oh, okay. Come on. Okay. And then let's go pull this up. And go in here. And now we're going to go to genecards.org. All right, so we want to make sure that we can actually see our genes. So just arrange it until you find it. It's okay. Now we're going to look up PRSS35 and see if there's a gene for it. There is. That's great. Now let's see if there's a summary. Okay. Predicted to be located in an extracellular region outside the cell. This is elevated outside the cell. It codes protein. Diseases associated with it. It doesn't have colon cancer listed. Well, that's about it. We'll go ahead and copy that. And we'll just paste it in. And we'll go to our next gene. Let's go ahead and make each row like 75 or 60 wide. Our next gene is CTAGE1. It looks like they have it. And it is cutaneous T-cell lymphoma associated antigen 1. It's predicted to be involved in endoplasmic reticulum. That's an organelle inside the cell outside the nucleus. And that is for packaging up stuff, endoplasmic reticulum. I believe it's packaging up proteins that are made by the ribosome in the cytoplasm so that they can be excreted from the cell into the body. It's either that or it's for destroying the cells. We can look that up though. But Golgi vessel mediated transport, Golgi apparatus transport, accretion, and vesicle cargo loading. So it has to do with packaging up a protein and making it a finished product to bring it to the extracellular space. And this other one was for extracellular. It's predicted to be active in the endoplasmic reticulum, exit site, and endoplasmic reticulum membrane. So we can just go ahead and look that up, but let's copy it first. It says diseases associated with clinical reticulatus. We'll go ahead and paste it here. And then let's see what the endoplasmic reticulum does. It's been a while since I... It's a continuous... Okay, it's an extensive network of membrane-bound tubules and flattened sacs. There's a rough and a smooth... Oh, yeah, the rough one has the ribosomes on it. RER is primarily involved in protein synthesis and folding. That's for the primary, secondary, tertiary, quaternary shapes of the protein that's made. Okay. Smooth one lacks the ribosomes, but the rough endoplasmic reticulum has the ribosomes. Okay, so the smooth, it doesn't say, it just says the endoplasmic reticulum. So we don't know if it's a smooth one or the, yeah, basically it's for, it looks like it's for protein building, packaging protein, finished product, making them into their shape. All right, I only have 2 of these left. Look at it. Kind of hurts my jaw. I don't know why it's weird. Usually that doesn't happen. But I do have this 2 thing going on. Okay, so our next one is SB2C. They have it. Okay. Predicted to enable transmembrane transporter activity. Predicted to be involved in several processes, including chemical synaptic transmission, neurotransmitter transport, and regulation of synaptic vesicle exocytosis. This is also with transportation outside the cell, or that could be in mitosis, the exocytosis part. Exoskeleton is in a cell too. It says though it's in the plasma membrane, and each cell has a plasma membrane. The organelles do too, and synaptic vesicle. Predicted to be active in dopaminergic synapse. And that's either the stress response or the rest response. They call them different things. I took pharmacology, but it's like the opposite. It's totally grammar related. So We are going to look that up. Active in dopaminergic synapse. Well, dopamine is a feel-good hormone. Whenever you don't have enough dopamine, you are said to have Parkinson's disease and a pale substantia nigra of the midbrain. When you have too much dopamine, you're said to actually be schizophrenic. Yeah. The synaptic vesicle membrane. Okay. And then here it says SB2C is a protein coding gene. Diseases associated with it. Intestinal botulism. Oh, that sounds crazy. Foodborne botulism. Uptake and actions of bacterial toxins and infectious disease. Transporter activity and transmembrane transporter activity. It seems to be another transportation gene. So far, we've gotten a lot of cellular transportation genes. Could be due to the fact that they are knocking out the AURKA gene. And so the way that they do it in vitro could be having to do with genes that transport these proteins. So we're going to go ahead and copy this and paste it into the gene summary for SV2C. And then PSG6 is our next one. Let's get rid of some of these windows because we'll have a whole bunch of them. PSG6. The heck? Didn't I just enter this? Hold on. Why is it? It's shutting out on me. Hold on. PSG6. Well, this one's pregnancy-specific beta-1 glycoprotein. This gene is a member of the pregnancy-specific glycoprotein gene family. The PSG genes are a subgroup of the carcinoembryonic antigen family of immunoglobulin-like genes and are found in a gene cluster on chromosome 19, Q13.1 to section Q13.2. Telomeric, the ends of the gene, the telomeres, keep it from unraveling. So another cluster of CEA carcinoembryonic antigen. That's an odd accumulation of words. Carcin, cancer, embryo, antigen. So it's a pathological substance to the embryo that's cancerous. That's what I would think it means. Telomeric. The PSG genes are expressed by placental trophoblast. That's a phase of forming the embryo from blastocysts in the early development, and released into maternal circulation during pregnancy, and are thought to be essential for maintenance of normal pregnancy. The protein encoded by this gene contains the arginine, glycine aspartate tripeptide associated with glycine results in multiple transcript variants and protein isoforms. Okay, so this is a pregnancy gene. We'll just copy that and paste it. And then our next one is UTP14C. I'm gonna drink some water. Jeez. I guess I'm the only person who's on it right now, so they're suspicious of it. They're like, who the hell is on my site right now? Damn it. There's nobody like that. Just kidding. Predicted to be involved in several processes, including meiotic cell cycle, cell regeneration in the mitotic process, 4 stages: anaphase, prophase, metaphase, and then exophase or something like that. Ribosomal RNA, I think that's what the R is, processing and spermatogenesis, located in cytosol and nucleolus. There is a little nucleolus inside the nucleus. The cytosol is inside the cytoplasm of the cell. Is a protein coding gene, disease associated. Okay, we got that. Ribosomal RO. So it did say ribosomes have 2 parts. There's subunits. So it did say that here's the small subunit. There's a large subunit and then there's a small subunit. So that it can fit, it latches on, and then it lets the codons go through to make the proteins, the triplet codons that are the amino acids. Okay, copy and paste. These are all the AURKs, the stimulated genes. So this is going on in colon cancer. The fold change of knocking out the AURKA gene over colon cancer cells. So they said that knocking out the AURKA gene allows the body to benefit from treatment to heal from colon cancer. So it looks like all of this stuff is working on getting proteins from the cell out of the cell, processing the cell, and then there's something for the pregnant woman as well. And then for building proteins. So everything's going on right now to build proteins and transport them outside the cell when they knocked out the AURKA gene. So this is being stimulated. And so let's see let's just unhide these and look at where it ends, where they start getting inhibited. Because we're doing the read count. So it starts being inhibited at number 14 right here. So we could go up to number 13, but at number 14, they start being inhibited. So then we'll see a difference. Let's go ahead and hide all these columns again. Oh, what just happened? Didn't I just say hide? Okay, this needs to be hidden as well. All right, so let's save this because we did do work to it. And then let's look up our next gene. Let's close these other windows. Otherwise, we'll have a whole bunch of them. PLSCR4. Did we search for it? Because if we did, I would have to select that. Okay, good. So this is a phospholipid scramblase 4. And then we're going to the summaries. Enables the CD4. CD4 is the T cell. So you have CD4, and there is a multihistone complex 1 and 2. 4 and 2 make 8. So that one goes to MHC2. CD8 and MHC1 go together. And there is a difference in what they do. CD4 tells the natural killer cells to go out and do work to protect. And the natural killer cells are part of your white blood cells, and those are the MEN, MEN, monocytes, eosinophils, and neutrophils. Now, Ben are the granulocyte cells. Those are the basophils, eosinophils, and neutrophils. So just so you know, eosinophils can go out and destroy a certain worm and viruses. Those will be elevated when you have a viral infection if you have a healthy immune system. Basophils destroy bacteria. Monocytes are an acute infection, and neutrophils are always high normally. And so if they're higher than normal but higher than monocytes, then that means that you have chronic infection. Okay, just so you know. All right, let's read this some more. Enzyme binding activity and phospholipid scramblase activity. The phospholipids. Phosphorus lipids. Okay, involved in calcium activated phosphatidyl choline scrambling and calcium activated phosphatidyl serine scrambling. Okay, so this is chemical stuff, doesn't really ring a bell. So the bilipid, plasma membrane, and then the phospholipids, phosphorus. Okay. Phosphorus is also an osseous structure. Phosphate trashing hormone is from your parathyroid, the back of your thyroid. If you have high calcium levels, you have low phosphorus. This is an inverse relationship. If you have high phosphorus, you have low calcium. Your muscles and skeleton need calcium and your nervous system. So if you're running low on calcium, it's because your phosphorus is high. I'm just trying to make a connection here. And then we have this calcium activated phosphatidylcholine. Choline sounds familiar from somewhere. Acetylcholine, phosphatidylcholine. Those are neurotransmitters, possibly. Scrambling and calcium activated phosphatidyl serine scrambling. Okay. Located in nucleus and plasma membrane inside the cell, enables CD4 receptor binding activity. So from this, I can say the CD4 cells need to bind to the antigen or have the antigen bind to it so they can send out signals to the natural killer cells to look for that and destroy it. Any cell with that marker needs to be destroyed so that it doesn't contaminate the rest of the body. Hold on. That's my work phone, but they know not to call me. It's a scammer. I said text. Yeah, I don't know who that is. That's not even like one of my alternate numbers or anything. I don't know why I get a lot of spammers. Okay. Okay, so we were over there. Catalyzes metal. The other definition. Okay. Located in nucleus and plasma. Okay, the other definition. A protein coding gene. Diseases associated with it. Atypical choroid plexus papilloma. It sounds like something with the stem cell of your brain. Or something similar. That was my last half an hour. We don't need to know that. Calcium ion binding and SH3 domain binding. I'm not sure what that is. There's a Uniprot definition. Catalyzes metal ion induced ATP independent rapid bidirectional and non-specific movement of phospholipids between the inner and outer leaflet of the plasma membrane. Participates in the redistribution of phospholipids between membrane leaflets. Metal ions binding to the calcium binding site and induce conformation change in the protein. All right. Something to do with phosphorus and lipids. And then I'm going to research. We're going to copy and paste that for immunity, CD4 binding. They knocked out the AURKA. This is still an upregulated gene. So now we're seeing some activity where he wants to go hunt for whatever is destroying the host. Because now they have fixed some kind of elevation stimulation of AURKA by having it inhibited. Okay, so our next gene is SLC24A3. I'll go ahead and close that. They have it, solute carrier family. 24. All right. Plasma membrane sodium, calcium exchanger are an important component of intracellular between the cells, calcium homeostasis, calcium balance, and electroconduction. Potassium dependent. So the sodium potassium regulation is 3 sodium out to potassium in the cell of a neuron, nervous tissue, so that it can conduct depolarization to send a synapse, a signal, a nervous signal to the brain or to the organ as a motor function or a sensory function, up or down. Okay. Potassium dependent sodium calcium exchangers, such as SLC24A3, are believed to transport 1 intracellular calcium and 1 potassium ion in exchange for 4 extracellular sodium ions. So between the cells, they transport 1 calcium and 1 potassium in exchange for 4 sodium ions from outside the cell. Disease-associated, developmental and epileptic encephalopathy, corneal dystrophy, posterior polymorphous, transport of inorganic cations. Cats are positive, so positive chemicals. Positive elements of the chemistry table. Anions are the negative ones. The negative, right? The positive. It's been a while since I looked at a chemistry table of elements. But yeah, I think the metals to the gases, so metals, the negative anions, positive gases, cations, and amino acids, oligopeptides, and visual cycle and retinal rods. So it seems to be part of the sodium, potassium, and calcium. Calcium for muscle activation, nervous tissue activation, contraction of muscles, smooth and skeletal, and heart as well. Okay, so we're going to copy and paste this. Okay. And then we're going to go to the next one, ZP4. These are all still upregulated. Zona pellucida glycoprotein 4. All right, let's see what this does. An extracellular matrix outside the cell matrix that surrounds the oocyte, the woman's shell for the sperm in making an embryo, from the cycle and early embryo. It is oh yeah, the zona pellucida. Yeah, that's the extracellular matrix that surrounds the oocyte. Okay, the zona pellucida. Yeah, I remember that from the anatomy developmental. Okay, so that is where the spermatocyte has to penetrate and go into, and then once there's one that reaches it, then it shuts off and it seals off, and nothing else can go in there. It is composed primarily, and so there's one embryo. It is composed primarily of 3 or 4 glycoproteins with various functions during fertilization and pre-implantation development. The nascent protein contains a nitrogen terminal signal peptide sequence, a conserved ZP domain, a furin cleavage site, and a carbon terminal transmembrane domain. So when there's amino acids, they talk about stuff like this, like the H group and then the N-terminal and C. It's been a while since I had that class too. It is hypothesized that furin cleavage results in release of the mature protein from the plasma membrane for subsequent incorporation into the zona pellucida matrix. However, the requirement for furin cleavage in this process remains controversial based on mouse studies. Previously, this gene has been referred to as ZP1 or ZPB and thought to have similar functions as mouse ZP1. However, a human gene with higher similarity in chromosomal synteny to mouse ZP1 has been assigned the symbol ZP1, and this gene has been assigned the symbol ZP4. Zona pellucida glycoprotein is a protein-coding gene. Disease-associated prostate cancer, ovarian dysgenesis. So agenesis is doesn't develop, and dysgenesis usually means it didn't develop all the way. Or hypogenesis as well. Among its related pathways are fertilization and meiosis, which is, so there's mitosis for cell division and replication of any cell in your body. And then there's meiosis, which is for creating another human or another mammal. So another life form from 2 different sex chromosomes. That's what that one is. So it's in fertilization and meiosis. Annotations include obsolete signal transducer activity and a crocin binding. Okay. Component of the zona pellucida and extracellular matrix surrounding oocytes which mediates sperm binding, induction of the ichrosome reaction, and prevents post-fertilization polyspermy. So that sounds exactly what I said, but in science words earlier. The zona pellucida, it allows only one sperm to get in. Once one breaks into it, it seals off, and then it doesn't allow any others to enter, so that there's one embryo. The zona pellucida is composed of 3 to 4 glycoproteins. Okay, so this is something to do with fertilization in meiosis. There's one other one with pregnancy. This one. So these two, UTP14C and ZP4, seem a lot to do with embryo pregnancy related stuff. Other ones are more with the cell transport, protein shaping, protein building, and then the conduction channels for muscle and nerve function. We're still on the upregulated one. Next one's RET. We'll just copy that and paste it. You know, it's like the shortest gene so far. And we'll go ahead and go to that one. So the RET gene, it's a gene encoder for a transmembrane receptor and member of the tyrosine protein kinase family of proteins. Binding of ligands such as GDNF, glial derived neuroreceptor. Glial. I can think of brain with that one. And other related proteins to the encoded receptor stimulates receptor dimerization and activation of downstream signaling pathways that play a role in cell differentiation, growth, migration, and survival. The encoded receptor is important in development of the nervous system and the development of organs and tissues derived from the neural crust. This has to do with the development, the neural crust, and developing the embryo. I think that is like the spinal canal, or else the spine for the central or peripheral nervous system. Yeah, everything's been a while, okay? So this proto-oncogene can undergo oncogenic cancer activation through both cytoplasmic genetic rearrangement. So rearranging the proteins that are made in the cytoplasm and activating point mutations Mutations in this gene are associated with Hirschsprung disease, which a little newborn gets, affects their gastrointestinal tract, large colon. It's basically dead on part of it, so the baby usually dies unless they get a surgery to fix it. And central hypoventilation, underventilation syndrome, and have been identified in patients with renal agenesis. They didn't have full development of their renal system. Renal, I think of kidneys, but could be the urogenital. Civic summary, and then the gene cards and a uniprot. Okay, so has more information. RET mutations of RET fusion lead to activation of tyrosine kinase receptor and are associated with thyroid cancers. Most point mutations are the most common mutations identified in medullary thyroid cancer, with germline somatic mutations, hereditary forms. Okay, and then the gene card one says Hirschsprung disease, that's twice. That having to do with the colon. The other one was with the thyroid. Oh gosh. Oh, it seems to have a lot of information, huh? Well, there's certainly a lot of information here. Okay, it shows up in many PubMed articles. Alrighty. We'll just copy this whole thing. I mean, I guess I could read some of this. Receptor tyrosine protein kinase involved in numerous cellular mechanisms, including cell proliferation, neuronal navigation, cell migration, and cell differentiation, in response to glial cell line-derived growth factor families. In contrast to most receptor tyrosine kinases, RET requires not only its cognate ligands, but also co-receptors for activation. GDNF ligands and first bind their corresponding GDNFR co-receptors, triggering RET autophosphorylation and activation, leading to activation of downstream signaling pathways, including the MAPK and AKT signaling pathways. Those are in cancer, usually breast cancer, they show it. Acts as a dependence receptor via the GDNF GFR81 signaling, and the presence of the ligand GDNF in somatotrophs muscle cells with somatotrophs within pituitary promotes survival and downregulates growth hormone, pituitary relation production, but triggers cell death, apoptosis, in absence of GDNF. So RET, it looks like, acts as a dependence receptor via the GDNF GFR signaling. It's a dependence receptor. In the presence of the ligand GDNF in somatotrophs within pituitary promotes survival and downregulates growth hormone production, but triggers cell death in absence of GDNF. Required for the molecular mechanisms orchestration during intestine organogenesis via the ARTN GFRA3. Okay, so we were dealing with colon cancer, and the last gene was having to do with Hirschsprung. Wait, this gene is it this gene? Okay, yeah, the last gene was Hirschsprung's. That was the first one to deal with colon cancer. This red gene is saying more about growth hormone and cell death. Oh, this one is Hirsch. This one is Hirsch. Okay. Hirschsprung. So this one is the first one with the colon cancer reference, but it's not cancer. It's just having to do with the cancer. And not having to do with the colon in infants. Okay. And then, yeah, the other stuff it said was like pituitary, pituitary gland hormones. All right, next one, IGSF10. Oh, it's making freaking cough. Throat's dry. IGSF-10 predicted to be involved in regulation of neuron migration. Predicted to act upstream of or within ossification, bone ossification, neuron migration. Located in an extracellular region outside the cell. IGSF-10, immunoglobulin superfamily member 10, is a protein coding gene. Diseases associated, gonadism, underdevelopment of gonads, with or without anosmia, anosmia, I don't remember what that one is. Primary ovarian insufficiency. So, underdeveloped reproductive system. Of early migration of may be involved in the maintenance of osteochondroprogenitor cells, osteochondrocytes for bone development. All right, let's copy and paste that. Still on the upregulated cells. And then we're going to go ahead and save this, but then we're going to actually go to this next one, SOX5. And this one, it used to be called the sex determining region. Why? Interesting thing in genetics, that region, if you have a lot of estrogen, and you can totally turn it into, if it was going to be a boy in early development, very early, I think it's like by day 7 or so, same as with an embryo for pluripotent cells. If you can have a lot of estrogen in the body by that time, you'll for sure have a girl, even if it was meant to be a boy, but it doesn't mean it's going to be a full functioning girl. Some of them are just underdeveloped males with reproductive systems underdeveloped, I believe. So they could be a female, but have male characteristics. Underdeveloped female parts too. Okay, that was just interesting. But this is what that gene is. It used to be the sex determining region Y. Okay. The summary is this gene encodes a member of the SOX SRY related HMG box family of transcription factors involved in the regulation of embryonic development and in the determination of the cell fate. The encoding protein may act as a transcriptional regulator after forming a protein complex with other proteins. The encoded protein may play a role in chondrogenesis. A pseudogene of this gene is located on chromosome 8. Multiple transcript variants encoding distinct isoforms have been identified for this gene. So this is like the third pregnancy-related one or embryo development one. It looks like it has to do, the last one had to do with the gonadotropin releasing hormone, right? So the pituitary gland. And that has to do with hormones as well. The developmental stages and fertilization also use those at certain times. Now we have another early development embryo gene. I mean, I am not aware if these samples in this commercial cell line are from a female. Maybe the female had colon cancer and was pregnant. I'm not sure why we are seeing so many genes for pregnancy, but it is a commercial cell line that they knocked out the AURKA. We can look that up and see. They did give information of what it is. So usually they, when we get through all the upregulated ones, which is just this one and another one, we're going to look up in the series information what is before we get to the downregulated ones. So, okay, chondrocyte differentiated cartilage formation. So that has to do with estrogen for the bones, or even androgens. chondrogenesis. This one, the sex determining region, but also cartilage formation, chondrogenesis. Okay. These. All right. Let's go to the other. Okay. Okay. Transcription factor involved in chondrocytes, differentiation, and cartilage formation specifically binds the 5' AAC-AAT to 3' DNA motif present in enhancers and super enhancers and promotes expression of genes important for chondrogenesis or the formation of cartilage, including cartilage matrix protein coding genes such as COL2A1, AGC1, required for overt chondrogenesis when condensed prechondrocytes differentiate in the early stage chondrocytes. So a lot of these tumors seem like they're made of cartilage because they're so hard and they're like a rock. This could be something forming cartilage, excess cartilage in a tumor. It's upregulated. But it's forming cartilage chondrocytes. All right. So together with SOC6 required to form and maintain a pool of highly proliferating chondroblasts between epiphysis and metaphysis. Long bones, they have a metaphysis is like the outer edge, epiphysis is the top, diaphysis is the long bone of it. So like if you're looking at like the, I believe this is the ulna, and then you have the little, this part here, and then the ulna has that olecranon process, I believe, right there. So the metaphysis would be the part that forms into that disc part. Epiphysis on top, diaphysis is the long bone. Pre-hypertrophy, but promote hypertrophy, and to delay terminal differentiation of chondrocytes in contact with ossification fonts. It seems to be a gene involved with cartilage formation around bones, long bones could be, or it could be the vertebral bodies or hyaline cartilage around those long bones or regularly shaped bones. Usually the hyaline cartilage is at the end of two interacting bones in a joint. So it says, yeah, and the chondrocytes are for the cartilage part. All right. So let's go to our next one. This is the last upregulated gene. It's IQCN. Let's close that other window out. And this one, it's involved in spermatid development, another fertilization gene located in mitochondrion. I've heard of mitochondria, but not mitochondrion. Implicated in spermatogenic failure, unable to create sperm. IQCN is a protein coding gene. Diseases associated, spermatogenic failure, 78, spermatogenic failure. It's essential for spermiogenesis and fertilization, creating sperm and fertilizing. May be required for manchete assembly and elongating spermatids. Okay, this has to do with the male reproductive system. We'll copy and paste. So there's like 4 or 5 fertilization embryo developments. I mean, it's possible that this maybe we're seeing these early development embryo genes from them using this on pluripotent stem cells of an embryo. I'm not sure, but we're going to check out what that commercial cell line is. So we will go ahead and go to our RStudio, and then we'll look at our series data because we have it here. We didn't erase it. Series 42, series 23. Okay, SW480 stably. Colorectal cancer cells of SW480. Let's go ahead and copy that. And we'll do an internet search. Let's go to this one. Okay. Okay. Cell line is a well-characterized human colon adenocarcinoma model derived from a Duke's C colorectal cancer patient. It is widely used in cancer research to study tumor biology, metastasis, and therapeutic responses. Source tissue, large intestine, morphology, adherent growth, disease, adenocarcinoma, Duke's type, satellite stability, key mutations. It doesn't say the gender, but maybe there's more information on it. Research applications, notable features. It exhibits phenotypic plasticity. Neuroplasticity means you can adapt. Phenotype is what's displayed from your genes, from your recessive or your inherited genes. Phenotypic, what you will show. Recessive or is dominant. And those are, I think they just call them genes, but when it says phenotypic, that's what it translates into, that protein, what you will show for it, your characteristics. I don't see any other information, but maybe okay, maybe this has information. Oh, so if you wanted some of these, you could buy them for $577 each. Learn about ways to use necessary Homo sapiens, human cells, doesn't say additional product information. I don't say who the colon was from. If you're into microscopy, it shows you. Alrighty. So it would be nice to know if this was a male or female, because, or, I mean, it's just weird that the adenocarcinoma colorectal tissue is showing reproductive genes with high fold change values when they knock out the AURKA gene. These ones seem to be high in expression for sperm creation, oocytes, development event. What was all of them? Okay. See, I mean, it would be nice to look at this table. Let's just open the other site. Okay. And see, just flag. I'm just going to highlight it. Did we save it as that? Okay, probably not. So, I mean, I can add another column and just say, look, this one is related to like reproductive stuff. And it's in the colon cancer cells that were treated with the AURKA inhibition. So I'll just put this one notes. Okay. This one was cell transport, right? All right. Let's go ahead and make this a little lighter though. Read more of it. All right. Yeah, I guess I got to go into the. Okay, that's the end of it. Or extracellular. I'll just put cell. And this one, cell. Okay, so this one's extracellular, like transport, right? Okay. Oh, it didn't say transport in this one, so I'll just say extracellular. This next one. That one is cell organelle, cell transport. Cell transport, organelle. Okay, and then cell transmembrane. Cell transport membrane. This one, reproductive, female, reproductive, male reproductive. So, oh no, this is the ribosome. So this one, ribosomal processing in the cell. I'll just put plasm. Ribosomal activity. I think this one was the endoplasmic reticulum. Endoplasmic reticulum. No, this one wasn't that, but it was in building the ribosome. I'm going to put cell cytoplasm ribosome. Subunit. This one was a phospholipid CD4 receptor activator. It's like an immune response, CD4 receptor binding, phospholipids. Okay. Plasma membrane, sodium, calcium, potassium, ions, like nerve muscle connection. It says it is a plasma membrane, sodium calcium exchanger. Plasma membrane, ion for nervous, skeletal. Okay. Female and male. But it's on the, it's a female reproductive. This one. Nervous system development, neural crux, early development, Hirschsprung's. Okay. So this is a neural crust, Hirschsprung's early development embryo. We can also, okay. Extracellular, this one is the underdeveloped reproductive. Okay, underdeveloped reproductive. Okay, that's next one. Cartilage, bone. Okay, cartilage, bone formation. IQCM, the last upregulated gene. This is the male reproductive. Okay, these other ones, let's save that. Don't show again. Okay. It's because I have it as a CSV and using it in Excel, and I didn't save it as an Excel work file. All of these other ones are going to be the inhibited ones. And it looks like how far are we along? Okay, I want to take a break, but I don't want to stop the video. It's just a couple minutes. Okay. I'll be right back. We're on it. I forgot she was in Mr. Okay. Let's get back to it. All right, it's an hour and a half. We're back. We got to do the inhibited ones. We found a bunch of reproductive ones and cell transport, nervous, skeletal muscle activation ions, gene. Those are the upregulated fold change whenever they knocked out the AURKA gene in colon cancer cells from a commercial line. That there's a lot of reproductive and then meiosis genes in there. It would be nice to know what gender the person was. We looked it up. There was no gender attached to those types of cell line. It was from adenocarcinoma, though. And it would be helpful probably to find out what AURKA is. So let's go ahead and go to AURKA. We're going to go to the gene cards site and then continue filling this out. But let's just look up what the AURKA gene is too, because we never even did that. And we didn't really need to. It's kind of explained. It says it inhibits the colon cancer treatment. So let's find out what it is. The protein encoded by this gene is a cell cycle regulated kinase that appears to be involved in microtubule formation and or stabilization at the spindle pole. So that is, I believe, in mitosis, whenever all the chromosomes get together and they line up in the center of, let's imagine a circle for the cell. They line up in the center. Have you ever seen that movie Witch, where all the witches come together and they're just like, I think it was that movie or something. I don't remember. Anyways, so it's a magnetic pole, and there's a lot of different magnetic movements in it. That's what made me think maybe it happens the same, similarly in meiosis, except that it's split. So actually, I was thinking meiosis, but they're very similar. Mitosis and meiosis. So the spindle pole, and that's whenever the chromosomes their DNA and then they form it in mitosis, and they just replicate everything again in that cell. The encoded protein is found at the centrosome. It's the movie The Witch. That was a weird movie. Encoded protein is found at the centrosome in interphase cells, metaphase, interphase, exophase. I think that's the last one because it's anaphase, prophase, metaphase. Been a while. Neuroplasticity is kicking in. It's building that up. Okay. A processed pseudogene of this gene has been found on chromosome 1, and an unprocessed pseudogene has been found on chromosome 10. Multiple transcript variants encoding the same protein have been found for this gene. Okay, so it seems to be something involved in mitosis, spindle formation, for cell division. So it is, we know that this study is knocking it out because it happens to interfere with successful treatment in colon cancer with chemotherapy. So it seems to be blocking chemotherapy ability. And chemotherapy is supposed to prevent mitosis because it doesn't want cell division because it keeps dividing cancerous cells. So the AURKA gene actually promotes cell division, mitosis, cell generation of that cell. And so when they knock it out, somehow that is showing that the cancer cell line is accepting the cancer treatment, which is to not have cell division. And AURKA seems to be something that promotes cell division. Yeah, so it's something they've worked with. And then you always got to try different things till you find the right thing. But it's in vitro. Loss of NLP from mitotic centrosomes and CDK. Mediated phosphorylation, removal of CDC6, annotation of this gene, transferase activity, transferring phosphorus containing groups and protein terminase kinase. Mitotic serine 309 kinase that triggers the regulation of cell cycle progression. Associates with the centrosome and spindle microtubules during mitosis and plays a critical role in various mitotic events, including the establishment of mitotic spindle, centrosome duplication, centrosome separation, as well as maturation, chromosomal alignment, spindle assembly, checkpoint, and cytokinesis. It is required for normal cell division and for the localization of, and these are all from different articles that researched it. Its effects. It regulates protein levels of the anti-apoptosis protein, a protein that prevents BIRC5. I think I've seen that in the nasopharyngeal cancer one too. They used JAK1, I think it was, to inhibit BRK5 so that they could see what happens in Epstein-Barr viral-associated nasopharyngeal cancer. And TP53 is everywhere. That is tumor. Protogene 53. I forget what the P stands for, but it's supposed to be a tumor suppressor. So that's in like most cancer studies. They look at that one because it's a biomarker for cancer activity. All right. Oh yeah, for cell division. Okay, so that we looked at that one. Now we're going to go into filling in the rest of the information for the inhibited genes. We only have the notes here, so we're just going to create the notes. We can go ahead and freeze the first row so that we know what we're doing, where we're putting this information. Okay, our next gene is CLEC18A. So this one, one of three closely related paralogous genes on chromosome 16 encoding secreted proteins containing C-type lectin domains. These domains bind to carbohydrates in the presence of calcium and may be involved in cell adhesion, immune response, and apoptosis, cell death. Alternatively, splice transcript variants encoding multiple isoforms have been observed for this gene. Okay, so it seems to be involved in cell adhesion, immune response, and cell death. And it binds carbs and when there's calcium. All right, let's copy the whole thing. Binds carbs and okay, it binds carbs, calcium dependent, and we're gonna go ahead and put in the top of this cell adhesion, immunity, cell death. Okay, that's our little notes on it. The next one is BAIAP2L2. So far, we've had every gene show up. That's good. Okay, so this one, the protein encoded is phosphoinositide and promotes the formation of planar or curved membrane structures. The encoded protein is found in RAB13 positive vesicles and at intracellular contact with plasma membrane. Okay, so that's new to me. Phosphoinositides, it binds it. It's part of planar or curved membrane structures. So, the encoded protein is found intracellular contacts with plasma membrane. It's a plasma membrane gene. Okay, so we'll copy this and paste it, but it seems to be like a plasma membrane gene. Plasma membrane. And I'll just put plasma membrane structure. All right, we'll just use that as a note. The next one is IgA SF21. This gene encodes a protein which has 2 immunoglobulin domains and is a member of the immunoglobulin superfamily. Proteins in the superfamily are usually found on or in cell membranes and act as receptors in immune response pathways. Immunoglobulin, I believe those are the antibodies that they have for like A through G. Okay, so the M, the G, the A, the D, I think I too. Oh, it's been a while. Okay. It seems to be immunoglobulins, immunoglobulins and the cell membranes. And then also in inhibition of synapses in the brain. Regulates synapses different. Okay. So we'll just copy and paste that. So this is immunoglobulins, maybe antibodies, IgE, IgA, IgM, antibodies, and presynaptic differentiation, regulated differentiation. Okay. Regulates inhibitory synaptic inhibition. I'm just going to put synaptic inhibition. Okay. And then we'll save this. Let's see. Got a few more to go. INHBE. This is inhibin subunit beta E. Transforming growth factor beta, superfamily of proteins, proteolytically processed to generate an inhibin beta subunit. Inhibins have been implicated in regulating numerous cellular processes, including cell proliferation, apoptosis, immune response, and hormone secretion. Maybe upregulated in endoplasmic reticulum stress. May inhibit cellular proliferation and growth in pancreas and liver. Inhibins and activins inhibit and activate, respectively, the secretion of phollotropin by the pituitary gland. Inhibits activins are involved in regulating a number of diverse functions, such as hypothalamic and pituitary hormone secretion, germ cell development and maturation, erythroid, blood cells, insulin secretion, nerve cell survival, embryonic, axial development, or bone growth, depending on their subunit composition. Inhibits appear to oppose the function of activins. Oh, Dallas, you need to behave. You need to behave, Dallas. Oh, that's a lot. Okay. We'll copy and paste. It seems to be something involved in multiple cellular processes and hormone secretion. Cell processes and hormone secretion. DNA 17 is our next gene. Or DNA I7. Dallas, shut your mouth. All right, for this one. Beta tubulin binding activity, microtubule binding activity, cilium, microtubule cytoskeleton. Sounds like all mitotic processes. Part of exonemal dynein complex. Cytoplasmic microtubule. Okay, so part of the cytoplasm. Cilia function. Okay, so this one inhibited. These are all inhibited genes too. This one has to do with the cytoplasm. Cytoplasm structure and cilia. It said cilia. So that could be the duodenum cilia that processes all the nutrients. But it could be their cilia in the cytoplasm. Let's go on to our next one. It's inhibited. RENBP. Close out some of these other tabs. Renin binding protein. Inhibits renin activity by dimer with renin complex known as high molecular weight renin. Interconversion of N-acetylglucosamine to N-acetylmenosamine. Okay. All right. Okay. All right. So some kind of process. It catalyzes a dimer out of renin. Usually the dimers are not good. Those are associated with diseases. But not always. That's just what you see in some pathologies. We'll just copy and paste this. We'll just say renin diseases. Inhibits renin activity. Renin inhibition. Okay. Renin. Let me just see what renin is first. I feel like it's the kidneys, but I could be wrong. Maybe so right now. Kidneys. Okay, kidneys. We got kidneys here. All right. And then the next gene, and we only have a few more, so don't worry. IL-2RA. Interleukin-2 receptor subunit alpha. Receptor alpha and beta. Common gamma chain. High affinity attraction. Homo-1 dimeric alpha chain. Result in low affinity receptor. Alternative splicing. Something coronavirus. Cytokines are proteinaceous signaling compounds that are major immune response. They control many different cellular functions, including proliferation, diversity, also, okay, so basically, like cellular processes. Okay. Immunity, cellular processes. We'll just copy all of this. Next one is CXCR1. Ooh. There we are. And then we'll go down the list. All right, protein coded, G protein coupled receptor. Interleukin. It's a receptor for interleukin 8. Inhibits embryonic oligodendrocyte precursor migration in the spinal cord. Dendrocytes are the arms off of a neuron. Actually, that's how you get neuroplasticity too. Those dendrocytes, they can reach out to other dendrocytes. If there's not enough on that nerve to access your location, your file storage of whatever information you're trying to find to connect, it borrows them, and then when it doesn't need it anymore, it gives it back, basically. it just says, here, take it. We're cool, like not. Motif. It's an immune gene. Interleukin 8. I'm gonna put that copy and paste of this. Okay, so it's an immune gene. Interleukin 8. Binding receptor. Okay. This is inhibited. This immunity is in this receptor for interleukin 8 is inhibited. These are all inhibited genes. So renin inhibition is inhibited. Okay, we'll go over the notes afterwards. Let's just get all this information first. Next gene is TREM2. And this one, membrane protein, forms a receptor signaling complex. Coded protein functions in response and may be involved in chronic inflammation by triggering the production of. Okay, alrighty, let's copy and paste this. Looks like a lot of information here, though, from UniProt. Signaling and cell activation, ligand binding. We'll copy and paste that. And then, so that we know what we're writing, it is a mediates signaling activation following ligand binding. It's a receptor signaling complex, cell receptor signaling complex. We'll just put that. And then the next one is new PR1. And we can go through some of this. Binding to amyloid beta 42, mediates microglial activation. I believe that's in the brain. Glial proliferation migration, apoptosis, cell differentiation, proliferation migration, apoptosis. That's what happens in the normal cell cycle of immunity response, cell division. Expression of pro-inflammatory cytokines, inflammation response. Cytokines. We should put cell receptor complex. Inflammation response. Cytokines, I've heard of that. And then we'll go ahead and look up our next one. NUPR. 1. Tiny keyboard. You change the location every once in a while. Just so they phase you out as a person. Okay. Enables DNA binding. New PR1. Just this one and one more, and then we'll go over one. Oh, it might have cut me out already. Done. 1 hour, 59 minutes. Hang in there. All right, let's see where we at. Okay. NUPR1. DNA binding activity and transcription, coactivator activity, DNA binding activity, transcription, coactivator, program cell death involved in nuclear protein gene diseases, transcription regulator stress signals into a program of gene expression that empower cells with resistance to the stress induced by change in their microenvironment. Microenvironment, time, TIME. Transcription regulator that converts stress signals into a program of gene expression that empowers cells with resistance to the stress induced by a change in their microenvironment. Okay, yeah. So it regulates transcription and copying DNA to make a protein that converts stress signals into a program of gene expression. So it uses your stress to create, to go after genes, to transcribe them, that empowers cells with resistance to the stress induced, to empower cells with resistance to the stress induced by a change in their microenvironment. So cellular processes also involved in cellular processes. It's a transcription regulator. Cell, it's in the cell. Cellular processes, and it is transcription happens in the nucleus, and it is a nuclear protein. Nucleus. Yeah, nuclear protein 1. Okay, last one. PRR20B. And this one has little information. It is one of 5 identical locations in a cluster on chromosome 13 section Q21.1. The long arm, section 21.1. The predicted protein is proline rich and contains several dopamine D4 receptor signatures and Prince domains. Related to dopamine. Oh, okay, it's related to dopamine. Okay. Dopamine, something that's related to dopamine. Several dopamine D4 receptor signatures. Signatures of receptors for dopamine. Inhibited. Dopamine receptor signature. All right. All right. So those are the inhibited genes. Okay, we'll close that window now. And then we'll go back to this. I don't even know. Oh, you probably couldn't even read it, could you? Okay. So now that's not what was expected to happen. We'll go ahead and we'll look at this. All right. So let's unhide these columns. And let's just look at, we're going to hide all of them except for the last 3. Because we wanted the counts one. All right. So now we'll go ahead and lower that. And then we'll compare this. That. Trying to slide this bar over. doesn't want to. All right. So these are the fold changes. Those are the genes. Let's look. Inhibition of cell transport organelle, cell transport membrane, extracellular. Come on, dude. What the hell? Oh, we're at the top. Okay. These are stimulated. Okay. Extracellular activity, cell transport of the organelles, the membrane, female reproductive process, cell cytoplasm, ribosome, creating proteins, immune response, CD4 receptor, immune responses, activate ions for nervous and skeletal tissue, a female reproductive process, a neural crust, Hirschsprung's disease related gene is also elevated, early development embryo, underdeveloped reproductive cartilage bone formation, and the male reproductive system. All those are stimulated. Now we're going to the inhibited ones when they knock out the AURKA gene. AURKA gene is responsible for promoting cell division. So the colon cancer cells didn't want cell division, and that's what the chemotherapy does because it was dividing the cancer cells. And so these are inhibited now. Cell adhesion, immunity, cell death, these are inhibited. Binds carbs, plasma membrane structure, inhibited. Immunoglobulins, antibodies, inhibited. Cell processes and hormone secretion, inhibited. Cytoplasm structure and cilia, inhibited, of course. All of these are inhibited. So the cytoplasm structure inhibited. Kidneys renin inhibition is inhibited. So renin function is stimulated because it inhibited a gene that inhibits renin function. Immunity and cellular processes inhibited. Immunity interleukin 8 binding receptor inhibited. So now they're having the chemotherapy work. It sounds like they have a need to suppress the immune system. So they have these cells in the colon cancer with treatment and with knockout of the AURKA gene to inhibit the immune response of inflammation. Interleukins. And then cytokines, of course. Cellular processes, transcription in the nucleus is inhibited. So it's starting to do the reverse of AURKA, which is to suppress cell division. And then the dopamine receptor signature, inhibited. So there may be dopamine in the body, but they can't activate it as much. So they're probably not very happy because dopamine is supposed to make you feel happy. And all right, cool. So that's it. I'm going to share this. I'll upload it. I'll just go ahead and add another column that says activity. And it's going to be if stimulated. And I'm just going to go down the line until it gets to 13. Or we could just look at the counts underscore FC. And then now we're inhibited. I know we could just scroll it down because it's just a small file though, so we don't really need to do that. Otherwise, you could just take this little piece and then scroll it. But I don't really feel like doing that. My mouse isn't really working anyways. That great. I'm gonna go ahead and save this. It's the summaries added. We're gonna go ahead and upload this to Google. And then I'll give a link in the our Markdown document, and I'll share that document with you on ARPEDS. So we'll go ahead and go there right now. And we're uploading a file. And it's going to be the summaries added. Import the data. It's the top colorectal top genes, 23 unique underscore summaries added. We're gonna put colorectal cancer AURKA inhibition. We'll share this. Oh yeah, I shared the file with you, but I never name it the same thing when I upload it. I always just give it a name right there. So I'll copy this link. Done. I'll post it here in the file, and I'll knit it and share that at the link in the details. We added the summaries, and you can get that file here. Cool. Well, thanks so much. All right, awesome. So now let's go ahead and go to KEGG. I'll go ahead and knit this, and then I'll share it with you. I can't really, let's see. Let's close this. Let's see how far my video uploaded. Oh, 81%, and it's but another hour on top of that. Okay, and it's only a like 2.5 hour video. Oh, all right. We're gonna go ahead and visit. Go. It's already there. I'm gonna go ahead and republish it. You have the same document. It's in the chat, but I'm gonna details when I end this. Update existing. And we're going to explore KEGG and all the systems of the body a little bit. And you can see we didn't really add anything to this, but we did add something to the data. So you can see the link here. Open it in a new tab. There we are. And the information we added is at the end because that's where we hid the columns and put that. So there you go. All of them are good genes to predict the class of colorectal cancer wild type or with the AURKA gene knocked out. We just got more of a intuitive analysis of this. We can add these genes to our data set with the full genes on the next part of our larger data. But for now, let's get things going. We can close this out. Let our computer run a little better. Okay. So now we're going to go ahead and go to KEGG. And here we are. And then let's look up ultrasound. Nothing. Okay, let's look up circadian rhythm. Some genes. KEGG genes. These display all. Reading, knockout, take component system. Let's just look at one of them. Okay, gene bank. It has a symbol. Knockout two component system. Circadian rhythm. And possibly knowing someone's body language or intuition on things. Okay, let's go ahead and look up vertigo. It looks like this is a knockout These are associated with circadian rhythm. I knew that because I looked at it before. If you go to the homepage and you can scroll at the systems of the body. Let's go to Epstein-Barr virus, though, just to see what genes there are. Because maybe there's some Epstein-Barr virus genes that are that might be in that data. All right. There's a lot of genes. No knockout, no knockout. Ooh, lots of them don't have a knockout assigned. I'm assuming the knockout is something that can take out that gene. Oh, we found a gene associated with it. Oh, here's a way to knock it out and inhibit it so that everything can function normally. Really? Yeah, there's quite a bit of them. EVV. Wow, there's a lot of them. Okay, that's a lot. Okay, I think I remember reading something. There's like 700 and something of them. All right, let's just check one out. The symbol or it's not in all uppercases, so it's probably just a viral. Oh, it's E. coli. E. coli virus. Oh, that's odd. Okay. Weird. Okay, so let's go to one of these other ones. EB2, EBV induced G protein coupled receptor 2, Epstein-Barr virus induced G2. Symbol is EBI2. A sequence, nucleotide sequence, the DNA, amino acid. These letters are 3 nucleotides long. These are triplet codons for amino acids. Okay. The motifs. All right, now let's go ahead and look at vertigo. Nothing. Yeah, there's nothing really there. It's something, I guess, that happens physically in connection, but they have to limit their diet. It would just be like a pain response, I guess, having to do with blocking either two of the ear, or compressing it, or that fluid that swishes around to give you balance. Because you have 3 directional tubes in the ear. Okay. Yeah, for the up and down, and then the side to side. And rotation. All right, so this is the homepage of KEGG. And then you can go to data and okay. Pathways, gene expression pathway enrichment association. This is where I saw that they had circadian rhythm. So the KEGG pathway, it says, knowledge of molecular interaction and relation networks for metabolism, genetic information processing, environmental information process, cellular processes, organismal systems, human diseases. Human diseases are where we're at right now. But then you can look at it like you're looking at a list or just a title page to like any book. Metabolism, it has the all of this information. So if you had any interest in that processes, if you wanted to compare like energy metabolism, lipid metabolism, and nucleotide metabolism, purines seen in people with gout. That's with alpha, and amino acid metabolism. So this is a lot of metabolism. You can scroll down. Biosynthesis. You got caffeine metabolism. Secondary metabolites. Aflatoxins are in mold on bread and almonds even. They can cause liver cancer and death. That's what we were told. And then other stuff, biosynthesis. Maybe you had something about a benzoate, like anything on like a Prop 65 if you wanted to see damaged by this. Xenobiotics, stuff from an animal. Cytochrome P450, that's on the electron transport chain of the mitochondria for generating energy, turning oxygen into that excess ATP for energy. And other metabolism information, genetic information processing. All right, a bunch of that stuff. Viruses. They got the, where is it? Antivirus, rotavirus, the stomach, HIV, the flu, coronavirus, entero gut virus, flavivirus, alpha virus. Those sound like mosquitoes. Ebola virus, hepatitis, the herpes viruses. So Epstein-Barr virus. And two is STD, and then there is one, as well as cytomegalovirus and shingles. Nobody ever really gets cytomegalovirus unless you're around a lot of dirty, infectious people and things, and poor hygiene, and plus immunocompromised. So if you're at risk if you had an organ donated to you and you're a recipient, because you have to knock out their receptors on the organ so that your body never tries to destroy it. And then also HIV too. And then also just aging and underdeveloped children and elderly, very tiny children and elderly are at risk to it. So there's we could check that out. And we can keep looking down the list. Environmental information processing. It goes into different species. Cellular processes. Cell death in different species. Eukaryotes, where eukaryotes are plants. Prokaryotes, those are the bacteria, I believe. I still haven't checked, but I'm pretty sure that's what it was, because we drew pictures. The immune system. The immune system, you can learn a bunch on this with all of these things going on. Maybe you can watch the Immune Sisters or something like that, or any other good immune pathway to explore the T cells and B cells and the whole process of immunity, because it's very useful for you to know, as it is how your body reacts to a pathogen and prevents infection and destruction. Prevents destruction. Human diseases, cancer, specific types of cancer, gastric cancer. we looked up the colon cancer, colorectal cancer, and then there's the gastric cancer. All right. Infectious diseases. Okay, so we got herpes simplex virus 1, human cytomegalovirus virus. That's in a herpes family. It's in the herpes family. So is Kaposi sarcoma. That's a herpes virus too. And that one is seen in also immune deficient people. Epstein-Barr virus. That's warts. I could kill you a good catcher of the female reproductive system. And also the gastrointestinal tract. Other bacterial infections, parasitic, immunity, neurodegenerative. Oh, we can look up multiple sclerosis, I think. Nope, actually we can't because it's not in there. That is odd. Parkinson's is there and Alzheimer's, but substance dependence, cardiovascular disease, fungal infections. All right, so this was quite a long video. I'm going to put that RPubs link in the details, but you get an idea of this. I saw circadian rhythm somewhere in there. I think it was in the body systems, maybe. Oh, there's metabolism. Genetic information. All right. Maybe. Maybe it was there. I'm not sure. Knockouts. Look at this. The KEGG. Oh, it's the KEGG orthology. I thought I meant knockout. The KEGG orthology database is a database of molecular functions represented in terms of functional ortholog. A functional ortholog is manually defined in the context of KEGG molecular networks, namely KEGG pathway maps, bright hierarchies, and KEGG modules, and is given a KEGG orthology identifier called K number. Most KOs are defined from experimentally characterized genes and proteins in specific organisms, which are then generalized to other organisms based on sequence similarity. The granularity of function is context dependent. The resulting KO grouping may correspond to a group of highly similar sequences in one organism. Okay. I think my throat is just from talking. That's probably why. Right. We can look at the mapping. Okay, so there's nothing there. We didn't find anything on ultrasound. We might as well look for autism. That was our last one. Oh, there is something for autism right there. Pathway. Let's look for pathway. Nothing in pathway. Genes, let's see. Oh, a bunch of genes, actually. All right. Well, that's it. Next time we'll add these to our project. Our overall project, our large database. It is something we'll take the bold changes to. We'll do it in R. Let's go to the desktop. Is the R programs, all pathologies. I think it's in this. No, it's not in that one. Oh, I didn't save it from the last one. That's weird. Wait, yes, I did. Current pathologies database. Our latest one is from April 5th. Gastric carcinoma. All right, so I didn't add This is where we added the large B cells. All right, it's probably in our last one, which is the okay, no, it's not that one. Okay. This one. Oh, I have to look at that one. Pathology, all. Okay, yeah. So we need to copy this one. It should have been in the other file, but I didn't do that. All right. So we'll go to the all pathologies, current pathologies. We'll paste it there. And it's basically this. And we have currently 305-165 was our last one. The Epstein-Barr virus and diffuse large B-cell lymphoma. Those are our genes. Now we're going to go ahead and yeah, that's right. We didn't add the folder because we couldn't, because we didn't have any actual full changes. So there's not a large database of it. Because they just gave us 3 different B-cell lymphomas. There's no baseline between them. They're all a lymphoma with similar characteristics, but not the same. Although the study said there's a transition state between it. All right. So we'll go ahead and call it a night. What a fun Saturday night, right? Okay, cool. Accomplished something. Oh, such relief, right? Okay, great. Well, have a great night, and I will put this link in the details so you can get it. And then from there, you can get the link to the other database.